All right, it's time for the last lecture of the day. Last lecture. All right, so in this last lecture of today, because I've recorded like 5 lectures today, we're going to look at an example of a disease for induced pluripotent stem cell treatment. Okay, and we're going to go through a little refresher on ipsc. We're going to go through the procedure on how you use induced pluripotent stem cells to treat disease or look at disease, study disease. How to steps, right? And then we're going to go through a reasonably in-depth example of how to use induced pluripotent stem cells to treat a particular disease. And that disease is I have it here, hypercardiomyopathy, right? HCM disease, example, right? And within that example, we're going to go through how to refine iPSCs, right? We're going to go through how to create and also know that you have iPSCs, because that's kind of important. We're also going to go through how to take those iPSCs and turn them into cardiomyocytes. From iPSCs, right? Then we're going to go through how you can use those cardiomyocytes, which you've taken from someone who's diseased with HCM, so hypercardiomyopathy. So you're going to take cells from a patient who has hypercardiomyopathy, induce pluripotent stem cells from the patient's cells. And then we're going to go through how you use those induced pluripotent stem cells to make cardiomyocytes, and then eventually use those cardiomyocytes to study the disease that the individual has, right? Right? And, this whole process will be reasonably in-depth, and we'll go through a fair amount of the nitty-gritty of exactly how to make the cells and the details of that whole process. So, let's get started with a schematic of stem cell therapy. Right? So step 1, you have your human person, right? You have your human person, and you take from them a sample. That's a disgusting drawing. Right, and in that sample, you're going to have a range of different cell types, right? You might have, some skin cells and some muscle satellite cells or some fibroblasts or, whatever. A couple of different cell types, right? And then we're going to induce those cells to become pluripotent. Induce pluripotent stem cells. Make sense, right? So we take those and induce pluripotency. Right. Now, as we may have discussed before, I think we have, in order to induce that pluripotency, we use something called Yamanaka factors. Yamanaka factors are the 4 pluripotency inducing factors that we use in order to take, say, a myocyte or a myoblast or a fibroblast and turn it into an induced pluripotent stem cell. I'm going to talk in a little bit more detail about the local environment that's required to make that happen. We're going to look at the day 7 embryo and compare that to the situation of your stem cell, of your culture, sorry. But that's the gist, right? And so, yep, that's cool. Hypertrophic cardiomyopathy, that's the right word. So then we have our induced pluripotent stem cells, and I'm going to do another colour, and we induce them to become, and I'm drawing these cells of arbitrary shapes just to delineate between them, but you know, we need to induce them to become cardiomyocytes, right? Now we need to induce them to become cardiomyocytes because that's the kind of cell that we want to study, because this is a disease of the cardiomyocytes. So we've got our cardiomyocytes, and then we can do 3 different things with them, right? And this applies to all stem cell therapy. There are 3 things we can do. So we can perform disease modeling. We can perform direct stem cell therapy. And we can also perform drug examinations, drug testing. And we're going to see later in this lecture how each of those plays into this particular disease of hypertrophic cardiomyopathy. So, the first step. Well, the second step actually. So the second step, you see that? Not really. The second step is taking your sample and inducing it to become pluripotent stem cells. Now, as we know, you need your MyoD, your OCT4, your SOX2, and your KLF4, right? So those are your transcription factors. Transcription factors activate different parts of the genome. They open up the genome because they're pioneering transcription factors, blah, blah, blah. In addition, we need to test whether or not the cells we get out are indeed induced pluripotent stem cells. Now this is very important because we need to be testing these things. We need to know that what we have is stem cells, otherwise we're going to be injecting strange things and testing strange things, and the results we get won't necessarily reflect to reality at all. And so there are a number of ways we do that. We test for markers. We test for correlation of gene expressions between embryonic stem cells and induced pluripotent stem cells. And if we find that our gene expression is, within a certain range, because, there's heaps of genes, if we find that our gene expression is within an acceptable range, we're all good. I think I need to draw things a little bigger. And so that's important, that we ensure that the expression of our genes is indeed correct for induced pluripotent stem cell. And then once we have induced pluripotent stem cells, we can induce them to become cardiomyocytes. Side note, when we're inducing our induced pluripotent stem cells, we're going to use those 4 transcription factors that I mentioned. Something that I didn't mention in the previous video is you don't want to keep feeding them to transcription factors, because if you keep feeding them with something that stimulates Wnt signaling, for example, Wnt signaling, WNT signaling, what you'll get is an overexpression, and you won't get any induced pluripotent stem cells at all, or cardiomyocytes at all. So that's the next step. All right, so now let's look at how to take those induced pluripotent stem cells and turn them into cardiomyocytes. Okay. So let's have a look at the 7-day-old embryo. 7-day-old embryo. Right. And the thing that we want to look at is the mesenchymal cell area, which is over here. Right, we take that out. Okay, So our goal when we're inducing our cardiomyocytes is to produce an environment that is exactly the same as the environment that they have when they're induced in the day 7 embryo, 7-day-old embryo. Right, so we have Wnt signaling, which goes into BMP4, which provides FGF2. And don't take my word for it, also watch the lecture on this. But we need to induce those pathways, and then we need to stop with the Wnt signaling because of the thing that I mentioned before, because if we overexpress Wnt signaling, we won't get any cardiomyocytes at all. Now there's a whole range of anatomy that we can go through with this exact area. But the important thing is that BMP4 has a lot at the top end and less down here, right? I always forget what the top and bottom things are called, and I don't care because I don't study anatomy, haha. So we get a lot of BMP4 down there and less BMP4 down here, which means that we can fine-tune the exact kind of cells we get depending on the expression, depending on the amount of our different factors, right? So with a lot of BMP4, we're going to get lateral, dorsal, not lateral, anterior, whatever. The top endy one, those kind, and then we turn it down, we get these kind, and we want these ones in the middle kind of deal, right? Seems pretty reasonable, I think. So, let me look at my notes for a second. Yep. Posterior and anterior. Pos and... yeah, so this is the primitive streak. That's the primitive streak. And it's during gastrulation that we look at this. And all mesoderm and endoderm tissues arise from the primitive streak, which seems to be important. Okay, and these little guys in here, which I encircled in black, these are the cardiac progenitor cells, right? Cardiac progenitors. Progenitors, right? Yep, cool. So our goal, and this is the important thing to remember, our goal when we're culturing our cells, which are outside the body, our goal when we're culturing the cells outside the body, because we've taken a sample from the patient's skin and we've induced pluripotent stem cells, and now we're trying to differentiate those pluripotent stem cells into cardiomyocytes, which means we need to get them through cardiac progenitors and then through further steps to get them into being cardiomyocytes. And we need to take into consideration the exact microenvironment to which they are exposed as they differentiate. And that's the importance of this slide here. Okay, next thing. I'm just going to show you a quick timeline of differentiation. A quick timeline. It's not that important, but it's, you know, on day 0, we have BMP4. On day 1, we have BMP F plus FGF. Is that beta FGF? Yeah. That's FGF2 plus also activin A. Activin A. So at day 1, we have both BMP, FGF2 and activin A. At day 4, or day 6, say, so day 6, we have VEGF and DKK1. FDKK1. And at day say 10, day 10, we have VEGF, DKK1 and beta FGF. or basic FGF, which is FGF2, which is FGF beta, which is that's it. Right? Cool. So that's just to show you the little cocktail of drugs that we need to use in order to differentiate these cardiomyocytes correctly. It's not that much, but it needs to be in very precise measurements for very precise amounts of time. Wunderbar. So, we've successfully differentiated our cells. Now we ask the question, do the cells that we've created have the attributes of the disease? Now, there are a number of ways to test this. We can do genome sequencing. We can look at the behavior of the cell and stick a probe in there and see if they are cycling calcium correctly or if they have action potentials correctly. But the idea is, and this is an essential step, we need to verify that each of the cells that we're testing, that the population of cells that we're testing, has the disease characteristics. The cells that we're testing must have the disease characteristics. Otherwise, what's the point, right? So we've taken the cells from our patient, we've refined them, we've induced them into pluripotency, we've created cardiomyocytes. We now need to know that those cardiomyocytes indeed have the disease. And that's a whole step of the profile, but it's kind of really neither here nor there. An important thing to note at this stage is that hypertrophic cardiomyopathy is expressed after 40 days in culture and on average up to 18 years in patients. So let's have a think about why that is. Why would we express a disease after only 40 days in culture after 18 years in human patients, right? And there are a few reasons for that, right? So number one, right, so we have like internal compensation. We have internal compensation, we have receptors that can pick up issues with the heart and other tissues of the body, and the rest of your body is going to try and compensate for what's going on. In addition, and this is the real reason, so if you're going to write one down, this is the one to write down, it's the tension on the cells. So when we culture cells, we culture them in a plastic environment, we culture them on a plastic growth sheet or whatever, and that's a very high strength, high tensile environment for the cells, right? A stiff medium with high loading. A stiff medium with high loading. And what that stiff medium with high loading reflects in the human is a human transitioning from childhood to adolescence. So an adolescent human. Because when you're an adolescent, you're going through a growth spurt, you need to have more blood pressure, you need to move more blood around. And so you have a much greater blood pressure. And so your cardiomyocytes are going under much greater strain, which is similar to the behavior under these plastic culture conditions. Which is why our cells behave after 40 days like they do after 18 years in patients. Okay? That's all, it's relatively straightforward, I think. Yep, that's cool. So, still getting there. So now we've verified that our cells in fact have the disease. We've answered a couple of interesting questions along the way. What do we do? What do we do with the cells? What do we do? What's the point of everything that we've just done? Okay, so there are a couple of examples that we can use to look at this. We can test drugs, we can do disease modeling, and we can also do replacement of tissues, essentially. We're only going to look at drug modeling and disease modeling. And so, the important thing to note about these diseases is it could be one or it could be several genes that cause it. And depending on the complexity of the genomic makeup of our disease, we might need to take a different approach. It may or may not be useful to do IPS for a given individual. And indeed, depending on the situation in terms of the drugs we can use to treat the disease, it might not be worth it at all. So if you have HCM and you go to hospital, you're likely to get treated with standard heart failure drugs because we really don't have a good way of treating it at this point in time. But in fact, through research, through investigating with induced pluripotent stem cells, we were able to determine that the cause of the disease was a difficulty in cycling calcium difficulty. And so if you can treat that pathway, if you can treat that calcium cycling difficulty directly, the cells are no longer dysfunctional. But that was unknown until we were able to trial a bunch of treatments on these cells and a variety of patients and perform genomic editing. And then finally, we took our cells in the petri dish and we induced a situation of very high calcium, and we got the same arrhythmia that we got with this disease. So that's the case with this particular disease. So let's take another example. Let's take another example of when you would want to do disease study in order to understand a particular disease. So there's a cancer drug called doxorubicin. And doxorubicin increases the survival rate of cancer amongst adolescents by like 80%. It's spectacular, it's really good. Amazing. But it also causes heart failure. Now we're going to use this example to illustrate two possible approaches. Now it causes heart failure. So if you want to know whether it's going to cause heart failure for a particular patient, what do? Well, you can go through the process, get your cardiomyocytes or get your particular cells that are going to be affected by your your cardiomyocytes, and then you can apply the drug to them. You can see if they get sick, and then you can get your outcome directly for that specific patient. But it costs a lot. It takes a lot of time. It might not yield the results that you want immediately, and doesn't really have that much benefit on a patient per patient basis if we were able to get that information, in fact, from genomic sequencing or something cheaper. So the alternative, instead of screening the drugs, is disease research. And so what that means is we can either use genomic-wide association modeling, association study, whatever, or IPS association study. Now, genome-wide association, or GWAS, we take the genomes of a whole variety of people who are affected, say, by deoxyribin. Heavily affected, a lot of toxicity, and low toxicity, and we see the genes that they have in common. The thing is that might yield about 100 different genes, or 50 genes, or it might only work with specific background, or it might only work for specific individuals. And it's a very large exercise. Like, to get statistical significance across the hundreds of thousands of genes, it takes a very large sample size, right? And so instead, what you can do is the same thing, except when we have an IPS population, we can correct genes and see if that fixes it. Gives you a confirmation approach. Okay, so that's pretty cool. Deoxyribin, yep, 5 mutations. And so that's those are the two options. So I'm just going to summarize because we got through this faster than I expected. So HCM was a disease that we didn't really know what was going on with. But through taking IPS cells and examining the way that they cycled calcium and the way that different genomes responded, we were able to A, figure out what the disease was caused by, which was calcium cycling, and B, induce a situation in which healthy cells had the disease by creating a high calcium environment. That allowed us to refine the pathway of treatment because we knew that it was calcium based. And then you can treat this particular disease with standard drugs. that was all achieved using IPS because we couldn't have confirmed that high calcium environment without the use of IPS. We additionally look at a cancer drug, and the cancer drug is toxic, but it's really good at treating cancer. So we need to know how toxic a given individual is going, how much of a response they're going to have. And so in order to do that, we can take their exact cells, test the drug on those cells, and see if they're going to respond particularly badly or fine or whatever. And you can vary the dose depending on that. So at the moment with this particular drug, we don't necessarily give the full dose to everyone because we're afraid that we might induce heart failure. If you could know that you weren't going to induce heart failure using IPS, obviously that's a good thing to do. In addition, there are challenging situations in terms of genomically based diseases, which can be assisted with IPS cells. So if we have a suspect gene that we can knock out or induce, we can do that in IPS, perform the same thing and see if you get the clinical phenotype. And if you've cured the clinical phenotype, you've cured the disease. And I think, yep. And that is our summary of IPS and disease in a dish. Thanks, guys.